Hi, my name is Allison Kohlberg, and this is the video for my Unit 2 case connections for medical biochemistry at UNE. The date today is November 24th, so let's get started. The first case we will discuss is case number 2, which deals with the regulation of glucose uptake in normal cells and tumor cells. The case is as follows. The above brain MRI shows a large glioblastoma tumor with areas of necrosis within the tumor. On the right is a PET scan from the same individual following administration of fluorescently labeled D-glucose. Before talking about the glucose uptake in the glioblastoma, it's first important to establish how glucose is regulated in healthy tissues. There are a few different mechanisms to control glucose uptake in the body. Glucose transport proteins, otherwise known as GLUT, transport glucose into the cell through its concentration gradient. These proteins and their expression reflect the levels of glucose metabolism in the cell. There are 4 different transporters: GLUT1, GLUT2, GLUT3, and GLUT4. GLUT1 is fairly tissue nonspecific but tends to be used mostly in erythrocytes. It is a glucose and a galactose transporter with high affinity. GLUT2 is found mostly in hepatocytes, pancreatic beta cells, in the small intestine and kidney. It is a glucose, galactose, and fructose transporter with high capacity and low affinity. GLUT3 is found in a variety of tissues, including brain and neural cells, and is a glucose and galactose transporter with high affinity. Lastly, GLUT4 is found in skeletal muscle and adipose tissue and is a glucose transporter with high affinity. As such, the following mechanisms regulate glucose uptake in the following cell types. In the liver, it's regulated by GLUT2, in skeletal muscle by GLUT4, in the brain, GLUT3, and in adipose tissue, GLUT4. Now, we can talk about the differences in glucose use in normal cells versus tumor cells. Normal cells have a lower glycolysis rate, which is preceded by oxidation of pyruvate in the mitochondria. In tumor cells, there is a significantly higher glycolysis rate, followed by lactic acid fermentation. This is described by the Warburg effect. In glycolysis in tumor cells, the glycolytic rate can be up to 10 times faster than in normal cells. This provides more energy and food to the cancer cells, which allows them to metastasize and grow quickly. Additionally, tumor cells are able to quickly convert pyruvate to lactate with or without oxygen present, so ATP production is faster. Another modification is that excess glucose is sent to the pentose phosphate pathway, otherwise known as PPP. This provides synthesis of lipids and nucleotides and helps the cancerous cells combat antioxidant responses from the body. The second case study we will discuss is case number 4, which is as follows. As a previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat other than breast milk since birth. The baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports these symptoms started. Laboratory results are consistent with hypoglycemia but are otherwise unremarkable. Epinephrine is administered and there is no increase in blood glucose, however there is an increase in blood lactate. Physical exam reveals a liver edge 4 centimeters below the right costal margin. Percussion of the right chest and abdomen confirms hepatomegaly. Blood glucose levels increase after breastfeeding, but she cannot maintain blood glucose levels within a normal range during fasting. These symptoms are definitely troubling for any infant. They all point to one likely diagnosis: glycogen storage disease, otherwise known as GSD. This is a genetic condition with several different types. However, type 1, otherwise known as von Gerk's, is the most common. Because the body is unable to properly store the complex carbohydrate in the liver, when blood glucose levels fall between normal feedings, there is not enough of a reserve of glycogen to maintain proper blood glucose levels. This would explain the hypoglycemia, or low blood glucose, and lactic acidosis, both found in her lab work. Lactic acidosis occurs because gluconeogenesis is impaired, which results in the buildup of lactate. It occurs in the vast majority of people with GSD. Because the liver is unable to properly store the glycogen, it can oftentimes cause hepatomegaly when the organ is larger than normal and inflamed, which is what was found on the physical exam. The other 3 options for diagnosis can be ruled out as follows. In hereditary fructose intolerance, there is a deficiency in the enzyme aldolase B. This would produce hypoglycemia if the baby were to eat sucrose, fructose, or other sugars. However, as the baby is still only breastfeeding, this can be ruled out. In galactosemia, the body is unable to properly process galactose, which results in an increased level of blood galactose. This would cause large amounts of sugar to build up in the bloodstream, which was not found in the lab work. As such, galactosemia can be ruled out. Lastly, glucose 6-phosphate dehydrogenase deficiency, otherwise known as G6PD deficiency, occurs when the body is either missing completely or there are very low levels of the enzyme glucose 6-phosphate dehydrogenase. This enzyme helps erythrocytes function properly, and in its absence, such as in G6PD, hemolysis occurs. This would have been found in the blood work as it often presents as hemolytic anemia. Additionally, G6PD causes an enlarged spleen, not necessarily an enlarged liver. Lastly, G6PD actually causes hyperglycemia instead of hypoglycemia, which was what was observed. As such, G6PD can be ruled out as well. The following references were used for this report.